ठीक है
स्क्रीन रेडी है Hello, good morning. Am I audible, all of you?
Good morning, ma'am.
Morning. Am I audible? Can I start with today's lecture? You had your internal exams. How was your performance? Are you happy with that? Did you really do well in the internal exams, you feel? No answers? Yes, anybody? Unmute and answer my question. Are you happy, satisfied with your performance? Akshata? Nobody is ready to answer. Huh? Okay. So today we are going to understand about the role of free radicals and antioxidants in the pathogenesis of maintenance of health and various diseases related to that. So it is according to your competency 7.6 and 7.7 both clubbed together. That is the topic for today's lecture that we are going to understand here. So the objectives for today's discussion will be, or the topic is we have to be able to define free radicals, name and different types of reactive oxygen species formed in the body that we have to enlist, outline the processes that lead to the generation of free radicals or what are the mechanisms of their generation in the body, understand endogenous and exogenous sources of free radicals, enumerate the damages produced by the free radicals, explain the scavenging system that exists in the body, that is free radical scavenger enzymes, different types of antioxidants, clinical importance of these, process of lipid peroxidation, importance of antioxidants, and then the role of lipid peroxidation in pathogenesis of cancer, metabolism, and atherosclerosis. So these are the points that we have to understand in today's lecture. So most of the leading cause of the deaths in the developed countries and developing countries, they are related to mainly, we can name them, that is diabetes, atherosclerosis, lung cancer. Next comes in the list is, so these types of the disorders, around half of the deaths that are occurring may be because of the diet related problem, and half of them are going to be caused due to the damage caused by the free radicals. So these are mainly the cancers and this cardiac diseases. All these things are ultimately due to the production and they are the consequences of the free radical damage that is going to take place. So here we are trying to understand today what are these free radicals, what is the term a reactive oxygen species mean. So this is what we are going to understand today. So products of partial reduction of molecular oxygen which are unstable and highly reactive can damage the tissues. All of you know that oxygen is necessary for the life. Without oxygen we cannot imagine the life itself. But this oxygen, when it is being partially reduced, it is going to be deleterious and it is going to be dangerous for our existence. So these potentially dangerous molecules are called as reactive oxygen species, and they are obtained from partial reduction of molecular oxygen, and these are the reactive oxygen species. So most of the reactive oxygen species are free radicals in nature. Now you know the term. What is the meaning of the term free radical? This is an atom or molecular species that contains one or more unpaired electron in its outer orbital, and it is capable of independent existence. So this is the meaning of the term free radical. And we have to remember 2 points about it. It is having unpaired electrons in the outer orbital and is capable of independent existence. So oxidation reactions ensure that molecular oxygen is completely reduced to water. So the free radical molecule can be shown here like this. So this is the normal oxygen with the complete fulfilled valencies and in the outer orbital. Whereas here you can see this is lacking one electron. So here it is the normal oxygen with all the paired electrons, and here a free radical is a molecule or molecular fragment that contains unpaired electron in its outer orbital. So because of this, the important characteristics of reactive oxygen species can be free radical. They are represented always with a superscript dot. They are highly reactive with short half-life, 10 raised to minus say 9 seconds to few minutes. So thus they have very short half-life lifespan. They have the capacity to generate reactive oxygen. Once one reactive oxygen species is generated, it can generate the other or convert the other molecules to oxygen species, and that is called as the generation of ROS by chain reaction. So it is capable of doing that. They are strong oxidants and hence they are always cytotoxic. This is important point. They can damage various tissues and biological macromolecules. But along with these toxic effects, sometimes it is beneficial also. So through the respiratory burst, it plays an important role in immune system. So these are the deleterious effects that is damaging various tissues and biological macromolecules. But an important point here related to this free radical is because they generate this through respiratory burst, it plays an important role in immunity. So you can see the notions of some of the free radicals. So this red dot will indicate the radicals. Superoxide anion containing this one valency. This is hydroxyl radical with unpaired electron. These are abbreviated and denoted with the superscript dot. Whereas these molecules, though this hydrogen peroxide, it is highly reactive and hence it is, though it does not have any free radical, it is considered under the category of reactive oxygen species. Same is the case with hydroxyl ion. So these are also considered to be this, though they are not having unpaired electrons, they are highly reactive and hence are considered as under the reactive oxygen species. So there are two types of these free radicals. One is oxygen species and the second is nitrogen species. So they are called as reactive oxygen species and reactive nitrogen species. So this is the list of reactive oxygen species. All of you can see these are containing this free oxygen radical in their structure. Now, as I have told, this H2O2 and singlet oxygen, they do not have free radicals, but because of their extreme reactivity, they are included in the groups of ROS. Now, these ROS and RNS, that is oxygen species and nitrogen species, are further subclassified on the basis of groups of compounds that they contain. So they are subclassified as radical species and non-radical species. So these radical species contain at least one unpaired electron in the outer orbital and they are capable of independent existence. Whereas non-radical species, they are not free radicals but can easily lead to free radical reactions with the living organism. Got the difference between these two? So radical species, they are having unpaired electron in the outer orbital and capable of independent existence. Non-radical, they are not non-radical free radicals, but can easily lead to free radical reactions in the or living organisms. So they are important compounds. So these are the radical species and non-radical species. These are the examples of both the things. So you have to remember about superoxide, hydroperoxide, hydrogen peroxide, hydroxyl, zinc, lipid peroxide, singlet, nitric oxide, peroxynitride. These are under ROS, that is radical species. Non-radical species include hypochlorous, hypochromous acid, ozone, nitrous acid, nitrosyl cation, dinitrogen trioxide, and aldehydes, peroxynitrides, etc. So they are the, now here some of the ROS with their half-life. Just you can see few seconds from 10 raised to minus second to few minutes they exist in our body. So nitrogen dioxide, nitric oxide, 7 minutes it is around. Nitronium ion. So this is, they are having short lifespan. These are the examples of them. Now how they are produced inside the body. So this generation is either by the endogenous and exogenous sources. So they are produced during as the intermediates in the many biochemical reactions. So free radicals and ROS are continuously formed during normal metabolic processes in the body. The subcellular organelles like mitochondria, peroxisomes, endoplasmic reticulum are the prime endogenous sources as the oxygen consumption is high in these subcellular organelles. So apart from that, the sources can be summarized like this: the cellular, enzymatic, and environmental sources. So the cellular are mainly these organelles, mitochondria, endoplasmic, and peroxisome. Enzymatic are the reactions catalyzed by the cytochrome oxidases, xanthine oxidase, and membrane bound NADPH oxidases. All these enzyme reactions contribute to the generation of free radicals. Next comes the environmental sources. So exposure to ionizing radiations and the environmental pollution is going to be the external source for these free radicals. So now during the cellular metabolism, how it is generated. So about 1 to 4% of oxygen that is taken up in the body is converted to free radicals. And these free radicals are generally produced during the normal oxidation of the foodstuffs, or that means the oxidation of the molecules, that is carbon, carbohydrate, proteins, lipids, etc. So continuously they are going to be produced in the body. during the metabolism of these macromolecules. And all this is going to occur in the mitochondrial membrane due to the leaks in the electron transport chain in mitochondria. Next comes the reactions which are involving the enzymes like oxidases, mainly xanthine, aldehyde oxidase. They are going to form superoxide anion and hydrogen peroxide. Next, in the macrophages, they produce nitric oxide from arginine by the enzyme nitric oxide synthase, and this also will give you an important antibacterial mechanism. So in mitochondria during generation of NTP, that is breakdown or oxidation of these biomolecules, oxygen will be converted to this to water molecules, and during this process there is leakage of oxygen radicals in the form. When two molecules of this oxide radicals combine together, they will be, it is called as superoxide, generation of the superoxide, and the enzyme will be the dismutation reaction, that is superoxide dismutase will lead to the formation of this superoxide. So, and then there is formation of the hydrogen peroxide. So these are the endogenous cellular metabolism that will form or that leads to the generation of free radicals. Now, in the mitochondrial membrane, the most important source of oxygen in vivo is the most aerobic cell, that is the mitochondria. So, leaking of electrons during the electron transport chain, all of you will be knowing or understanding what is this electron transport chain. Maybe you have come across this in your earlier schooling. So, during the transfer of this electrons through the complexes of respiratory electron transport chain, from jumping from one to second, second to third, fourth complexes. During this period, when there is leaking of electrons during electron transport, that is going to lead to the formation of the oxide radical. So oxygen with the electron will form this free radical oxide. So the sequential univalent reduction steps of oxygen can be shown like this. So ultimately oxygen will be converted into water. So this molecular oxygen is reduced to water. But during this conversion, these electrons are taken up from one complex to the other, and during this jumping there is leakage of this electron, and that will lead to the formation of these radicals as the intermediate in the production of the ROS, that is reactive oxygen species. So oxygen, molecular oxygen will convert into this superoxide. Then this superoxide is converted to hydrogen peroxide. Hydrogen peroxide will be converted to this hydroxyl radical, and this is going to be converted to the water molecule. So this is the major contribution of the generation of superoxide free radicals or reactive oxygen species. So superoxide anion radical is the most important ROS formed by enzymatic processes, auto-oxidation reaction and non-enzymatic electron transfer reactions in which electron is transferred to molecular oxygen. So different complexes and enzymes are going to be involved in generation of this reactive oxygen species. Now in the macrophages, generation of ROS takes place through the NADPH oxidase, which is present in the inflammatory cells, that is neutrophils, eosinophils, monocytes, and macrophages produce superoxide anion. So you know this NADPH is mainly contributed by HMP shunt pathway. So this NADPH will be oxidized by this oxidase enzyme, which is given by these polymorphonuclear cells. This will be converted into the superoxide. By the enzyme SOD dismutase, it is converted to H2O2. Myeloperoxidase, in presence of chloride, will form the hypochlorous acid, and this is going to be helpful for the killing of the bacteria. So this is whole process is called as the respiratory burst, which helps in the phagocytosis. So absence of, now because of lack of this NADPH oxidase enzyme system, it will prevent the formation of ROS and results in the chronic granulomatous disease, that is the disorder related to the lack of this oxidase system. This leads to the formation of granulomata in many organs, and this disease is called, also known as the Bridges-Good syndrome or Quay syndrome. This is related to lack of NADPH oxidase system present in the phagocytes. So this is how the phagocytosis takes place with the help of this NADPH oxidase, and this is called as the respiratory burst. That is the advantages of free radical generation. Now in the endoplasmic reticulum, the reactions that are taking place with the help of oxidases, for example, xanthine oxidase, during the purine catabolism, this xanthine and hypoxanthine, they are converted into uric acid, and this will be going to really lead to the formation of this oxygen radical. Apart from that, another redox cycling by the oxidized cytochrome and reduced cytochrome P450. These are the detoxification reactions. They are also going to lead to the formation of the superoxide. Next is hydrogen peroxide, another free radical, this reactive oxygen species. This is formed in the cell by dismutation reaction spontaneously during respiratory burst or by the enzyme superoxide dismutase. Next is hydroxyl radical. They are highly reactive oxygen radicals, and they are formed in the biological system by ionizing radiation reaction of metal ions with hydrogen peroxide, that is by the Fenton reaction, formation of hydroxyl radical from ozone, and reactions of hydroxyl radical through hydrogen atom addition and electron transfer. So this is the Fenton reaction where the H2O2 combines with the salts of iron, leads to the formation of a free radical, this hydroxyl radical. This is another that is directly free oxide radical with H2O2 will lead to the formation of the hydroxyl radical. So this is Haber-Weiss reaction. So these are the two important reactions in generation of hydroxyl radical. Now apart from that, during this synthesis of the prostaglandin, there is production of the free radicals, and that is going to be the mechanism for pro-inflammatory regulation. Next radical that we have to understand is the nitric oxide. So now this nitric oxide is synthesized from arginine by the nitric oxide synthase. So there are different types of the nitric oxide synthesis. So it can be inducible, that is INOS, constitutive INOS, and NNOS, that is neuronal and endothelial. So these are the different types of nitric oxide synthesis. They will be converting this L-arginine to the citrulline and nitric oxide. So this nitric oxide, in the low levels, it is going to be very helpful. It is by the constitutive nitric oxide synthase, that is INOS. So this helps in vasodilation, antiplatelet aggregation, and neurotransmission. Whereas in the high levels, it is going to be toxic. It is because of this inducible nitric oxide synthase, and this is going to be toxic. So you know this significance of nitric oxide synthesis. This is going to act as endothelial relaxing. This nitric oxide is an endothelial relaxing factors in the low amount. So it is going to be having vasodilation effect, and this is synthesized for arginine from arginine to during conversion of arginine to citrulline by the endothelial nitric oxide synthase ENOS. So this is generation of this nitric oxide. Now this has got the role, it is used in the treatment in the form of nitrates, long used to treat the angina. So that is because of from these nitrates we get the this nitric oxide, that is the vasodilator, and hence it is used in the treatment of angina. So these are the various actions of this nitric oxide free radical. So this nitric oxide is a key messenger in the cardiovascular system. It stimulates growth hormone production, helps in immune system. It improves the memory and nerve cell plasticity. So it has got wide roles. So this is an important free radical that we have to remember, and also you have to remember about the nitric oxide synthesis, different types of the nitric oxide synthesis, that also you should know. So there can be a question of short essay on these different radicals and the enzymes involved in their production. Next is the peroxynitrite. This is produced by the reaction of nitric oxide with superoxide. So generation of one oxide radical will give or lead to the generation of the other. That is the chain reaction, what we have understood or learned in their characteristics, that one generation of one radical will lead to the generation of the other types of the reactive oxygen species. So this is how nitric oxide combines with superoxide and leads to the formation of peroxynitrite. And this peroxynitrite, because of its oxidizing properties, can damage array of molecules in the cell signaling layer, including DNA, proteins, and it helps in or results in the apoptosis, that is the cell death. So via this Caspa's activation, this will lead to the apoptosis. So there are various mechanisms by which this cell death occurs. So this is a signal for the cell death. So it is formed by this peroxynitrite. So nitric oxide will be converted by action of this superoxide, which is coming from this NADPH oxidases. So this will be converted into peroxynitrite. Peroxynitrite can activate Caspas. This can lead to lipid peroxidation and lead to generation of protein oxidation, protein nitration, inactivation of enzymes. Cell necrosis will take place this way. This is via mitochondrial activation. So this will lead to Caspa's activation and apoptosis, and cell death can be there because of the effect of peroxynitrites. So we can summarize the generation of endogenous sources of free radicals by these different metabolic reactions: leakage through electron transport chain, production of H2O2, xanthine oxidase, and this enzymatic conversion due to membrane lipid peroxidation, peroxisomal generation, prostaglandin, nitric oxide, phagocytosis, oxidation of heme to bile, auto-oxidation of different metal ions, that is by Fenton and Habermas reaction. Next, other sources are the environmental effects, that is during drug metabolism by the cytochrome P450 reaction, that is during detoxification of the drugs, due to damage caused by the ionizing radiations, photolysis, photoexcitation of next environmental factors like cigarette smoking, alcohol promotes the lipid peroxidation. So external sources can be classified as the environmental effects. So they can be due to drug metabolism, damage due to UV and X-rays, and cigarette and alcohol. So these are the factors, environmental factors, through which we are continuously exposed to the free radicals that are caused inside the body. So we have to take proper care while handling or utilizing these radiations and all. And these industrial pollutions by use of pesticides, herbicides, excess consumption of alcohol, smoking, consumption of high-fat foods, and environmental pollution, all these are going to be the external factors that leads to the formation of free radicals inside the body. How and what, that it is going to be through UV light, it is going to be converted into generation of the free radicals through mitochondria, white blood cells, air pollution, cigarette smoking. All these are going to damage our DNA, and ultimately there is going to be deleterious effects on the body. So now let us understand what are these harmful effects of these free radicals. So these free radicals can damage the biomolecules in our body. So it will cause the oxidation of sulfhydryl groups on the proteins, modification of amino acids. These reactive oxygen species damage protein by fragmentation. Aggregation results in loss of biological activity of the proteins. Then on lipids, it is going to be acting on the polyunsaturated lipid molecules of the membrane and are particularly susceptible to damage of them. That is because of the uncontrolled lipid oxidation that is called as lipid peroxidation. This on carbohydrates, it will cause the glycation that increases the susceptibility of proteins to the attack of free radicals. So glycation of the proteins will lead to the susceptibility of proteins to free radicals. Nucleic acid, that is on nucleic acid, it will lead to the formation of DNA strand breaks. Fragmentation of bases, deoxyribose results in cytotoxicity and mutagenicity. So these are the ways by which it can damage proteins, lipids, and DNA and RNA. So these are the biochemical groups which are involved. So sulfhydryl in protein, polyunsaturated fatty acid chains, and these are the hydroxy groups. So it has got role in the loss of function due to damage of proteins, membrane damage, permeability transition, and ultimately DNA damage, cell death, mutation occurs. This can be due to formation of 8-hydroxy-DG. On DNA, it will have the defect, and this 8-hydroxy-DG damaged DNA will be going to be toxic to the body. So, uncontrolled, if they are not taken care at the early stages, then it will lead to the diseases which are related over the period to the excess loss caused by the hyperactive free radicals and the diseases which are caused due to these free radical or the pathogenesis of various diseases like cardiovascular, cancers, inflammatory, respiratory, cataract, aging. All these things are going to be due to the damage caused by the free radical. So the free radical damage can be attributed for the diseases which we come across are due to the harmful effects of the free radical. So how in the cardiovascular diseases it is going to be contributing for the free radicals can contribute is through the oxidation of the LDL. So oxidized LDL formed by the action of the free radicals promotes the cardiovascular disease and atherosclerosis. Cancers is mainly because of the damage to DNA, which causes mutation, cytotoxicity, and that is the basic key role in the carcinogenesis. Inflammatory diseases, that is again due to damage on the extracellular components such as collagen, hyaluronic acid, that promotes glomerulonephritis and ulcerative colitis. Respiratory diseases, which is due to destruction of the endothelium and lung edema, that is triggered by cigarette smoke, which contains free radicals, and that promotes the formation of the lung diseases. So, now the mechanism how this oxidized LDL will lead to the this cardiovascular disease can be explained. So when the macrophages take up oxidized LDL, they when overloaded with the lipid become foam cells. These conglomerates of foam cells form fatty streaks or yellow patches visible in the arterial wall. And these dying foam cells release lipid that form the lipid pool within the arterial wall. So this is how these free radicals damage the LDL and this oxidized LDL form the foam cell, that is and there will be plaques of cholesterol, or you can say lipid plaques in the arterial wall that will block the blood flow. So this oxidized LDL will have these effects, that is normal artery, in the normal artery it will form the fatty streak, fibrous plaque, complicated plaque, and then it will lead to ruptured plaque. And because of this, unstable angina, stroke, critical ischemia, myocardial infarction, and coronary death will be the consequence, and it is because of the free radicals. Even in the perfusion injury after myocardial infarction, that is recurrence of again toxic effect on the myocardial ischemia is seen caused due to the generation of the free radicals. Now, the pathogenesis of diabetes mellitus also can be attributed to free radical through destruction of islets, result in the islets of the Langerhans, that is the production of insulin is hampered, and that can lead to the pathogenesis that can be cause for diabetes mellitus. Cataract again is due to free radical damage. Male infertility, due to this damage, it is seen due to reduced sperm motility and viability. Aging process is because of the exposure to, or throughout our life we are getting this free radical damage to the biomolecules, and that leads to aging process. So changes that are seen in the aged people, that is wrinkling, sagging of the organs, all these are age related process are attributed to free radical damage. Apart from that, the Parkinson, Alzheimer, multiple sclerosis, liver cirrhosis, muscular dystrophy, all these age related diseases are caused due to free radical damage. So this free radical toxicity affects all the organs: eye, air tube, abdomen, body, lower abdomen, chest, brain. So all these are seen due to aging. And now coming to understand what is this lipid peroxidation. So this lipid peroxidation refers to the oxidative degradation of the lipids. It is the process where free radicals steal the electron from the lipids in the cell membrane, resulting in the cell damage. So the unsaturated chains of the lipids, they take up this electron from the process whereby free radicals steal the electron from the lipids, resulting in the cell damage. So from these lipids of the membrane, the free radical takes up the electron, and that leads to the lipid radical production. Once the lipid radical is formed, there will be formation of the peroxidation, or the process continues via propagation and elongation. So the initiation, propagation, and ultimately termination, these are the three steps in the chain reaction of lipid peroxidation. So this process proceeds by the free radical chain reaction. It most often affects the polyunsaturated fatty acids, and ultimately there is generation of these lipid peroxide radicals, that is called as the lipid peroxide, and this lipid peroxide is going to be mutagenic and carcinogenic, and this is called as the lipid peroxidation, the consequences of which are mutagenic and carcinogenic. So now the body takes care of these toxic free radicals. So we have the mechanism to scavenge these toxic free radicals which are generated during the metabolism. So to overcome that, we have the defense system against these oxidants, that is called as the antioxidants. These are the body's scavengers of free radicals. As we can see, bacterial infections can be treated by the antibiotics, the damage caused by these oxidants can be overcome or reduced by the antioxidants. So these are the superheroes in the circulatory system. So body takes care with the help of antioxidants. So if there is imbalance between the generation of free radicals is more than the antioxidants that are present in the body, the state is going to be oxidative stress. There is going to be the state in the body that is called as oxidative stress. Now what happens is this normal cell, when it is continuously being attacked by the free radicals, it is going to be the cell with the oxidative stress. Now this can be controlled with the help of the antioxidant, and hence always inside the body there is a balance between the oxidant and free radicals generation and counteraction by the antioxidant, and that is the normal health. Under the normal healthy conditions, this balance is always maintained. But when the production of free radicals is more than this antioxidants, it is going to be going to be the situation of oxidative stress that leads to oxidative damage to the biomolecules like lipid, proteins, nucleic acids, and they are going to cause the tissue injury and inflammatory responses. So healthy cell is going to be attacked by the free radicals continuously, and the cell will be in the stress of oxidation, that is called as the oxidative stress. So this is now what happens depending on the severity of oxidative stress, the biological consequences are seen. So severity of oxidative stress is shown on the left side, and the right side you have the biological consequences. If the severity is less and gradual, it will lead to, this is what is happening during the aging. Then if it is medium and rapid, it is going to be carcinogenic and mutagenic. Large and rapid, it is going to cause the death, stroke, trauma, ionizing, due to it, ionizing radiation, etc. So these are the, during aging, it is slow, gradual decrease in the antioxidant status. This is more, if there is decrease in antioxidant, more and more free radical production will lead to carcinogen and mutagenesis. So we have the defense against oxidants in the body. They are called as the superheroes fighting against the free radicals, and they are the antioxidants of the body. So what do they do? So this is the healthy atom with no unpaired electron, where this is the free radical with the unpaired electron. Now these antioxidants, they scavenge these free radicals by accepting this, highly neutralizing their effect of this um, oxidation state. So the substance present in low concentration relative to oxidizable substrate that significantly delays or reduces oxidation of the substrate are called as the antioxidants. So they reduce the effect of dangerous antioxidants by binding together with these harmful molecules, decreasing their destructive power, and they can also help in repair damage already sustained by the cell. So they may be considered as the scavengers of free radicals. So antioxidants are scavengers of free radicals. They reduce the effect of oxidants by binding together with these harmful molecules. So during the reaction of antioxidant sacrifices itself by becoming oxidized. So inside the cell, this is going on harmoniously. Antioxidant status is there. Always these oxidants are being scavenged with the help of the antioxidants, and thus it prevents the membrane, nucleus, everything is protected by the presence of these antioxidants in the body. However, antioxidant supply is not unlimited, as one antioxidant molecule can react only with the single free radical, and hence a constant need to replenish antioxidant is required. So these antioxidants will prevents the transfer of electron from oxygen to organic molecules, stabilizes free radicals, and terminates free radical reactions. Now, there are different types of the antioxidants depending on their location. According to their location, they are called as plasma antioxidants, ascorbic acid. These are the various vitamins what we are learning in some of the, while understanding some of the fat soluble vitamins we have come across the term antioxidants. So they are these vitamins are acting as a intracellular antioxidants. Next is we have learned the role of vitamin E in the as an antioxidant. Next, intracellular oxidants are the enzymes like superoxide dismutase, catalase, glutathione peroxidase. So these are the different types of the antioxidants according to their location. So plasma, cell membrane and intracellular oxidant. According to their nature and action, they are enzymatic, non-enzymatic. So enzymatic are SOD catalase, glutathione peroxidase, glutathione reductase, etc. Non-enzymatic include nutrient antioxidants like beta carotene, alpha tocopherol, ascorbic acid. So the different vitamins, vitamin A, vitamin E, we have studied them. So these are the nutrient antioxidants, and this is the ascorbic acid. Metabolic antioxidants include the end products of certain metabolism, that is bilirubin obtained by catabolism of heme, uric acid by purine catabolism, the proteins, metal binding and transferring protein, ferritin, ceruloplasmin, transferrin, albumin, glutathione, all these are the metabolic antioxidants. So now these each enzymatic important enzymatic antioxidants like superoxide dismutase present in every cell in the body, which actually represented by a group of metalloenzymes, which with various prosthetic groups. It is going to occur in three forms. So isoforms of SOD are copper zinc SOD, manganese SOD, and copper SOD. So their locations are cytoplasm with two subunit, mitochondria, that is manganese, and extracellular is copper. So this SOD is the first line of defense to protect the cells from injurious effects of the superoxide. So this is the enzyme called as superoxide dismutase. Next comes the catalase. So when H2 hydrogen peroxide is generated in large quantities, catalase is used for its removal. So H2O2 will be acted upon by catalase and it converts it into water and oxygen. So this catalase is an iron dependent enzyme present in all the body organs, especially concentrated in liver and erythrocytes. Brain, heart and skeletal muscles also contain low amounts of this. So this catalase will convert hydrogen peroxide into water. So this is second important enzyme. Next is glutathione peroxidase. This glutathione is the selenium dependent enzyme. Entire process is driven by energy production at the cellular end, which involves proper thyroid hormone, healthy mitochondrial function, and an active pentose phosphate. So this is the role of glutathione in scavenging the free radical. Next, nutrient antioxidants, that is vitamin E, the most important lipid-soluble antioxidant present in all the cellular membrane. It protects the cell against lipid peroxidation. This vitamin E in brown tissue, this adipose tissue, prevents the peroxidation of the membrane phospholipids and avoids the cell damage to its antioxidant. So this tocopherol will take up this radical and it will become, along with vitamin C, it can act as a scavenger for free radical. So this next is the ascorbic acid that we are going to learn in next classes. Water soluble, present in citrus fruits. It is a chain breaking antioxidant and as a reducing agent in electron donor, scavenges free radicals and inhibits lipid peroxidation. It helps in the regeneration of alpha tocopherol. So in the previous cycle, so this tocopherol is regenerated by ascorbic acid. So both these two act in coordination with each other. Beta carotene, that is vitamin A, most important. It is composed of two molecules that we have seen. It helps in quenching of the singlet oxygen. This quenching of singlet oxygen is the basis of its well-known therapeutic efficacy in erythropoietic porphyria. Lycopene is responsible for color of certain fruits, and that lycopene also acts as an antioxidant. Alpha lipoic acid, another vitamin-like compound, it is produced in the body, helps in the recycling of other antioxidants like ascorbic acid, alpha tocopherol, and glutathione. So, like this, these are the other, in addition to the previous vitamins and all. Other important nutrient antioxidants can be CoQ10, selenium, proanthocyanins, catechins, quercetin, ellagic acid, etc., which are obtained from these food sources. Next, metabolic antioxidant important, glutathione. In addition to its role in substrate glutathione redox cycle, scavenger of hydroxyl radicals and singlet oxygen. It also helps as an important role in xenobiotic. Then these metabolic antioxidants are uric acid, ceruloplasmin, drug, albumin, transferrin, albumin, bilirubin, haptoglobin. So how this uric acid acts? This scavenges singlet oxygen and hydroxy radicals, inhibits iron and copper dependent lipid peroxidation. Transferrin prevents iron catalyzed radical formation. Albumin scavenges radicals on its surface, protects albumin bound free fatty acid from lipid peroxidation. Haptoglobulin bind to free hemoglobin and prevent the acceleration of lipid peroxidation. Apart from that, drugs which are taken, so there are certain pharmaceutical agents which are used for the treatment of certain diseases like gout and all, which is the xanthine oxidase inhibitor, that is allopurinol, folic acid, they are going to be antioxidant in nature. NADPH inhibitors, albumin inhibitor of redox cycling, statins are going to be the antioxidant. So if body's own internal antioxidant defense system, including superoxide, catalase, and glutathione, is sufficient, so normally cellular homeostasis is a delicate balance between production of free radicals and our antioxidant defenses. So always this should be high on, they should be on the top, that is antioxidant system should be on the top of this pyramid. That can be through these various sources, minerals, protein, flavonoids, carotenoids, glutathione, and that will primary internal antioxidants complete the defense system. SOD catalyze glutathione. Secondary dietary is, but are normally plays a buffering role. So these internal nutrients will play the buffering role. So always it is said that supplementation with drugs is never as good as supplementation with food. This is a Chinese saying. So all these antioxidants we can get from our diet through various sources. Another recent addition to the list of these sources of antioxidants, the green tea, all of us are hearing, most of many of you must be practicing. So that is the consumption of green tea. This is because of its antioxidant property. So this green tea is consumed in China, Japan, Middle East, North America, and nowadays even in India, many people are following or using this green tea, and that is attributed to its antioxidant property. So this is more powerful. It is said that it is around 200 times more powerful than vitamin E. It helps in scavenging free radicals at higher rate, reduces the risk of heart disease, lowers the LDL oxidation, prevents red blood cell breakdown, protects against digestive and respiratory infections, prevents cancers of colon, pancreas, and stomach. So these all diseases are mainly caused by the Oxidative stress caused by free radical and that can be overcome by consumption of these dietary inclusion of green tea. So we can say that for a healthy tomorrow we have to think what we should consume and what we should not. So to counteract the harmful effects of free radicals should not we should not smoke. Do not overdo your exposure to sun. Don't consume alcohol. Don't consume foods containing trans fats. Cardio exercises. Consume antioxidant-rich foods and use a good antioxidant formula. Keep your stress levels down. So, thus, in today's class, we have understood what are the free radicals, how they are produced, what is definition, properties, difference between ROS and RNS, generation, reactions, endogenous and next external sources that will lead to generation of free radicals, harmful effects of free radicals, how to counteract the effect of these free radicals, how the body takes care by the antioxidants, different types of antioxidants like enzymatic, non-enzymatic, and the metabolic. Now, how to assess the body's oxidative stress? It is done by the use of chemical methods for the detection of the biomarkers of the oxidative stress, and most of these biomarkers they are due to they are produced by damage of lipids, proteins, or the DNA itself. So the major biomarkers are lipid peroxidation, which generates the malondihyde and hydroxynonenal. So these are the biomarkers for lipid damage, oxidative lipid damage. So we have the methods for estimation of these levels of malondialdehyde, that is MDA, and hydroxynonenal HNE, and other 2-propanol, that is acrolein. So by these, if the increased levels of MDA are seen in the patient serum, we can definitely come to know that oxidative stress is the underlying cause for the pathogenesis of the disease. Next is protein cleavage can be detected by cross-linking inhibition of enzyme activity, altered immunogenicity. And this 8-isoprostaglandin F2 alpha or F2 isoprostane is the most reliable assessment for oxidative stress status for new risk for coronary disease. Glutathione, so they are the damaged markers, whereas the assessment of the enzymes we can come to know if their levels are normal, they can be the markers for oxidative stress. So reduced glutathione disulfide we can know for the assessment of oxidative stress. Apart from that, 3-nitrotyrosine, that is the essential for nitrated protein diseases. This marker can be used. Carbonylated proteins, that is protein carbonyls, that will help in oxidation of amino acid side chains. So these are the markers for protein and amino acid. And the DNA breaks can be known by the assessment of 8-hydroxy deoxyguanosine. So you should know at least what are the markers for the oxidative stress. Detailed methodology and all the aspects are not there for you. Only the thing how you can come to know whether the body is under stress or not. We can assess by the assessment of these biomarkers related to lipid, protein, and DNA. Apart from that, you can know the levels of these antioxidants present in the body. So you have to check both MDA as well as vitamin, this ceruloplasmin, vitamin C, vitamin A. If possible, if you do this, you will come to know about the oxidative stress and antioxidant balance of a person, and from that we can conclude whether it is the damage related pathogenesis can be the oxidative stress or not. So we have the methods for estimation of vitamin E in the lipid phase, vitamin C in aqueous phase, ceruloplasmin can be in the extracellular fluid, Apart from that, caffeine, cysteine, glutathione, vitamin A, beta carotene also can be assessed for the knowing the antioxidant levels in the patient. So overall, we can say these are the contributing factors. On the left hand side, whatever you are seeing, UV rays, radiation, pollution, stress, poor nutrition, all this is going to lead to free radical. This free radical generation is going to cause cancer, it will affect the aging, heart attack, diabetes, dermatitis, other health complications and so on. So this is the damage caused by the free radical and oxidative stress on all the organs, and hence this is an important topic for the pathogenesis. Oxidative stress as the underlying cause for the pathogenesis of several diseases. For you, mainly you have to concentrate on free oxidative damage in the pathogenesis of diabetes mellitus, atherosclerosis, these things you and aging, how it is going to affect. So that is the most important part for you as per your syllabus. And now from this topic, you can have all the types of the questions can be expected, and mostly they will be in the long, short essay and short answer type. Each of the individual enzyme, antioxidant enzyme can be asked as a short essay, short answer question. Then these in general broad category and free radical, oxidative stress, antioxidant, and individually short notes for 5 marks can be asked. Like, and pathogenesis of the diseases like diabetes, atherosclerosis can be the again short essay question. So from this, the take home message from the topic that we have learned today, which is an important topic for the health, normal health of the human beings, we have to remember always we have to go for the consumption of polyphenol rich fruits, vegetables and beverages, which is going to be beneficial to human health, as these antioxidants are found in fruits, vegetables and tea. So these contain the flavones, isoflavones, flavonols, catechins and phenolic acid. So all they can act as antioxidant, will prevent apoptosis, cancer and the inflammation in the body, which is the underlying cause of several diseases. So always consume these antioxidant rich foods that you can see. So think before you take the anything as a food. So our food should be the, not drugs should be our food. Our food should be full of these antioxidants. So thank you.